Welcome back to our channel. And today we will be starting one of the most important chapters from biotechnology, which is biotechnology and its applications. In this lecture, we will be covering the following topics, which are tissue culture, totipotency, micropropagation, and somaclones. Many direct NEET questions are asked from this portion. So keep your NCERT with you and beside you, and let's begin understanding these topic. So the first question which arises for us is, why do we actually need biotechnology in agriculture? Now, this question in today's year or in today's era, like nobody would be trying to ask themselves, because the biotechnology has mostly been implemented in our agriculture. But before, in the 1990s, the biotechnology was still new and getting implemented in the agriculture was still a big thing. So let's first understand the problem. So the first problem which we were facing was the world's population was continuously increasing. Yeah, it was continuously increasing. So now we know that as our population increases, we can see the following, that as our population increases, more people equal to more food. This is a very basic understanding, we can say, or a basic knowledge, that if there are more people, more food is being required to fulfill, and more food is required to complete all the nutrition and hunger of the people. And because of this, as more people are there, more food is required. Along with that, need for high agricultural production. So these all were, we can say, directly proportional to each other. More people required more food, and to create more food, we were in need of higher agricultural production. So because of these problems, there were mainly three approaches that were being used for increasing food production. We'll discuss these topic in a concise manner. The first one is agrochemical based agriculture. Second one is organic agriculture. And the third one for us is genetically engineered based agriculture. So these three were actually being used mostly to, and we can say is currently still being used for increasing the food production. So let's first discuss our agrochemical based agriculture. So in the sorry, in the agrochemical based agriculture, what is being mostly used by farmers is we can say is fertilizers, insect insecticides, third one pesticides. And by using these three tools, the results, or we can say the advantages, being shown by the farmers with were the following, which is higher yield of crops. But along with the advantages, there are several disadvantages which were also being seen by using the agrochemical based agriculture. The disadvantages were, first of all, it was very expensive. As we know, the chemical based fertilizer, insecticides, pesticides are very much costly, and farmers are not financially able to purchase them. Second thing which was being caused because of these chemical based tools were environmental pollution. And the third problem that was being faced was soil degradation. Due to the high use of these chemical fertilizers, insecticides, and pesticides, the soil quality was getting degraded day by day. Then comes the second approach which was being followed was, which is also still getting followed, is organic agriculture. Now, in organic agriculture, the tools we can say, or the yeah, we can mostly say that tools or the stuffs that were being getting used by the farmers were, first one is the compost, second is manure, third one is biofertilizers. Now, using these three tools by the farmers, the advantages or results which were seen by them was, the first thing which resulted from the agrochemical based agriculture was, it was environmental friendly. But still, organic agriculture came with one of the disadvantages, which is, due to using this organic agriculture, we saw lower productivity, which results in the low yield of the crops. Now, to resolve this issue, we come to our third crop based agriculture, which is the genetically engineered crop based agriculture. Now, in this, the tools that were being used were actually the tools which were being developed by scientists so that we can get a higher yield of crops, better resistance to the insects and other abiotic factors such as we can say the temperature or the soil or some other factors. And the third thing was reduced pesticides dependency. So what we're being mostly used in the genetically engineered crop-based agriculture, I would like to highlight it uses biotechnology that were being developed by the scientists to genetically improve crops. Now, because of this, many benefits were being noticed. So let's discuss those benefits again in detail. The benefits that were being seen was higher yield of crops, better resistance to abiotic factors, and reduced pesticides dependency. And this is actually what our modern biotechnology aims to achieve. So these are the three mainly crop-based agriculture that were being used and are still being getting used in some extent. But the famously one which are being used all over the world is genetically engineered crop-based agriculture. Now, we come to our second most important topic in the biotechnology and its application is actually green revolution. Now, before biotechnology came, the green revolution was there. Now, the green revolution successfully tripled the food production. Green revolution successfully tripled the food production. How? Like, how did it manage to do? It was managed to do through better crop varieties, irrigation, better irrigation system, fertilizers, and pesticides. Okay, but there was still a problem. Even though food production increased, population growth was continued. So food production increased, which we can say is directly proportional to higher population being seen. Also, but still there was one more problem. Conventional breeding became slow and yield improvement reached limits. Now, beyond that, the crops cannot be improved genetically, or we cannot increase it nutrients. Therefore, scientists needed a faster method. Because of this, scientists came across the very important thing in the biotechnology, which is the tissue culture. Now, this is a very important topic actually, guys. Now, what is tissue culture? I'll write the definition. We can understand it together. Tissue culture is the growth of plant cells, tissues, or organs in artificial nutrients media under sterile conditions. Now, what do we understand from this is actually tissue culture is, we can say, is one of the process or method in which we are trying to grow the plant cells, tissues, or the organs in artificial nutrients. Those are not the natural nutrients such as soil, water, or anything. These are artificial nutrients that are being created by the scientists under sterile conditions. Sterile condition means there must be no bacteria, virus, or anything present in the room, so that this tissue culture takes place. Now, because of this tissue culture, there is a subgroup, we can say, which was being noticed, and definition is being given is of explant. Now, explant is what? Any part of a plant taken out and grown in a test tube. Example, we can say leaf, root, stem, meristem. Any plant part can potentially act as an explant. We take the part of a plant, for example, we take the leaf, or we take the root, or we take the stem, and we use the tissue culture process to grow it in a test tube in an artificial, by using the artificial nutrients media under sterile condition. Now, after explant, we get the following important topic, which is totipotency. I know it sounds a little bit funnier, but still, that is the biological word being given. Now, the definition of the totipotency is the ability of a single plant cell to develop into a complete plant. Okay. So totipotency is actually a term that is being used for the ability of a single plant cell which which develops into a complete plant. For example, if we take a single plant cell of a carrot, it can be developed and turned into a full carrot plant. Okay. Now, just to remember it, what is the use of totipotency? We can use the following trick. Totipotency, we can break it down into two. Toti and potency. Totipotency means we can break it down into two factors just to remember it. Toti means total, potency as we know from English grammar, which is power. So totipotency equal to total power. And also totipotency, like total power about what? Total power or ability of a cell explant to regenerate into a complete plant. Okay. Now, why is it possible? Every living plant cell possesses the genetic information required to produce a complete plant. Thus, one cell can produce into a complete whole plant. This property is called totipotency. Now, the important factor which is being required to understand that how these all biotech process takes place in a sterile condition along with the artificial nutrients, we need to also understand what nutrients mediums are required. Nutrients medium requirements. Okay. For plant growth, the medium must contain the following. We can break it down and write in a table format. Carbon source, inorganic salts, vitamins, amino acids, and last but not the least, are growth regulators. Now, for carbon source, we have the following example we can say is sucrose. For our growth regulators, the two mostly which is being mentioned in the NCERT, our NCERT is auxins and cytokinase. Remember these names. This is very important actually. It is being mentioned in the NCERT. Now let's move to our another topic, which is micro propagation. Now in micro propagation, the definition is as follows: production of a large number of plants through tissue culture. Okay, so why is this important? Look, from tissue culture, for example, we were being successful in creating or producing one single plant. But is that enough? No. What we need? We need it in a huge number. We need it into thousands or lakhs. So to produce that, micropropagation comes into place where production of a large number of plants through tissue culture takes place. So now, as we discussed, it has the following advantages, which is rapid multiplication and commercial production. It rapidly multiplies the whole plant for us, and it can be used for the commercial production. Now, for commercial, now some of these examples are, whichever NCERT mentioned, are tomato, banana, apple. Okay. Now, our last topic for this YouTube part one for the biotechnology and its application is actually soma clones. Okay. Now, what is soma clones? Actually, soma clones are the plants that are produced through tissue culture. Okay. Now, for example, in biological term, through tissue culture, we reproduced one plant. Okay. So now by using the micro propagation, we, if I write here, micro propagation, we were able to reproduce multiple plants. These multiple plants are called soma clones. Why? Since they originate from the same parent, they are genetically identical and same parents, right? Such plants are called soma clones. Okay, so soma clones is this thing. Yeah. So now there is like nothing more to be discussed from our end. Now in the next video lecture, we'll be checking the, the more topics where we'll be also covering the other topics of the biotechnology and its application. Yeah, before going, I would just like to um provide you some of the names which I just examples which I just remembered for the nutrients medium requirement is, for carbon source we have sucrose. For inorganic materials, for inorganic salts we have the following minerals, which are our nitrogen, potassium, phosphorus. Okay. For our metamins are being used to help in metabolism. Amino acids are being used to help growth and protein synthesis. And our growth regulators are being used for like the auxins used to promote root development, cytokinin cytokinins used to promote shoot development. So yeah, these are the main topics about the biotechnology. And I would request you all to go through all of these definition and in a little bit detailed manner through your NCRT, and we'll meet in the next video. Until then, goodbye.